Okay, let us begin by examining individually the 4 macromolecule groups. I'm going to split up 4 lectures, each lecture 1 group. First group we're going to discuss are what are referred to as the carbohydrates. Okay, when you see carbohydrates, think sugars. Okay, the carbohydrates are going to be found grains, fruits, vegetables. Now, yes, there are going to be some carbs, minimal. We'll see when we talk cells that are going to be found in animals, but nowhere near what's found in plant life. Now, what's great about these is that carbohydrates can be broken down into glucose. So, the majority of carbohydrates you're going to find in plants are going to be long strands of glucose molecules, all joined together using that dehydration reaction. Now, monosaccharides, this is going to be our monomers. You take 2 monomers, put them together, you get a disaccharide. That's just 2. Disaccharide, trisaccharide. After that, the numbers just become ridiculously large, measured in the billions of glucose molecules. We refer to these as the polysaccharides. When it comes right down to it, most of the monosaccharides you're going to be looking at, you're going to be dealing with in biological systems, are going to have 3 to 7 carbons. Okay, it's not always glucose. There can be others. And since I have told you many times already in lectures, shape dictates function. Even the shape of the sugars dictates how and what it can be used for. You look at where's the carbonyl group. Remember, carbonyl, COO. CO, right there, C double bond O. Is it terminal or is it somewhere within? Glyceraldehyde, dihydroxyacetone. Look at them. C3H2 H8O3. Same number of carbons, same number of hydrogens, same number of oxygens. But where that carbonyl group is, terminal, interior, different shape, means that glyceraldehyde, dihydroxyacetone, the aldose, the ketose of that 3-carbon sugar are going to be used completely different in biological systems. How many carbons are present? 3, 5, 6. Triose, pentose, hexose. They look relatively similar, but glucose, we're going to see in a later chapter, will be split. It's going to be split right there to form 2 glyceraldehydes, which will then be turned into pyruvate. And again, isomers come into play. We're going to see that for some metabolic functions, most of the enzymes that we're going to see are basically just rearranging the furniture. Glucose, galactose, fructose, relatively the same, but hey, look, where's the carbonyl? Aldose, aldose, ketose. Difference between these 2 guys? Right there, look at that. Look at that. Where's the hydroxyl pointing? It makes a difference. One's an important source of energy that we readily use, and galactose, part of lactose. Take lactose, cut it in half, glucose, galactose. It's the milk sugar. We can use it, we can readily use it, but not every organism can. They're not interchangeable. Now, you are in biology. You are not in a chemistry course. If you are going to draw the geometric shape, if you're going to draw anything, any sugars here in biology, whether it's this course or any of your upper division course, you have to keep in mind, we down here in biology only deal in things in an aqueous solution. Upstairs in chemistry, they deal with things in the theoretical. They deal with things in different contrived solutions. Everything in your body, everything in your dog, your cat, the tree outside is in a water solution, an aqueous solution. So what happens is the straight chain that we saw on the previous slide circularizes. Instead of a straight chain, it becomes a geometric 6 or a 5-sided sugar. So everything here is geometric. Nothing here is straight chain. So for our sugars, individual monomers, glucose, fructose, it's going to come together and you're going to get that dehydration reaction. The dehydration reaction that's going to give you what is called a glycosidic linkage. Ding, 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 ding. Focus right there. Glycosidic linkage. You ever see that term? That means there's an oxygen that is holding two monomers together. You're only going to find this in sugars. I'm going to go ahead and give you a heads up. All four of the classes of macromolecules going to be talking about, each one is going to have its own characteristic bond. Glycosidic linkage is the special bond found when water is removed between two sugar monomers that are then joined together. Now, it's not always going to be like it shows here, where the oxygen is below the plane of the two sugars. Sometimes it's going to be the two sugars are on different planes, or different things are going to be occurring. Where's the hydroxyls? What's pointing up? What's pointing down? Which sugars are being joined together? But it is always a glycosidic linkage holding two sugar molecules together. Now, when you get into polysaccharides here, as I said at the beginning of this lecture, can be anywhere in the millions to billions, billions of sugars long. They can be one continuous straight chain, or it could be branching chains, kind of like a vine. Starches. Starches are nothing more than a way for plants to store sugars. Remember, each of those sugars is going to be an energy source. Took energy to link all those carbons together to make the sugar. You break them down, it's going to give you energy back. Turns out the starch you're going to find in plants going to come in two forms: amylose and amylopectin. Amylose, unbranched, straight chain. What they're trying to show here in this long rope configuration is that this is going to be long chains of glucoses. Takes energy to link them together. That's when you break them down, and then you go through and you start breaking apart each individual sugar itself. Gonna get energy back. Amylopectin is a mixture, straight chains as well as branches like vines. Different shapes, different functions. Both, though, are used for storage. So one's more long-term, the other's more short-term. Then you can also take these polysaccharides, string them together in an alternating up-down of the glycosidages. linkages, below, above, below, on and on, over and over, and you get what's referred to as cellulose. Cellulose is not a means of storing sugars, storing energy. What cellulose is a form of structural. Cellulose is what allows trees, allows plants to stand up above the ground. Great example of cellulose. Anytime you go and you eat celery and you get that little string stuck between your teeth, that's cellulose. That cellulose is billions upon billions of sugars stretched together, and then each of those strands wound around others to form larger and thicker fibers. It's structure. The same cellulose that allows your celery to stand up, that gets caught between your teeth, is what allows sequoia trees to stand 200-300 feet up in the air. Lastly, when it comes to our polysaccharides, the hard outer shell of insects, that exoskeleton, composed of what is referred to as chitin. Chitin, as you see right here, long polysaccharides. Long polysaccharides allows these insects, allows arthropods to make and maintain and replace their shells by just what they eat. They eat vegetation. They get the starches, those sugars, which will then be linked together via dehydration reaction to form chitin, a new shell.